Thank you very much. I'm going to be telling you about the research that we've conducted here in Mississauga for the last 10 years. And I want to tell you about medicinal chemistry and how we've used medicinal chemistry to target proteins that are involved in cancer and how we've developed new therapeutics to target those proteins and kill effectively cancers. So I'm going to show you how we design and make small molecules to bind to proteins. And this binding event leads to cancer cell death. And in my time, we've been developing inhibitors, drugs, of a protein called signal transducer, an activator of transcription 3 protein, which is a bit of a mouthful, so we call it STAT3 for short. As you can probably tell from my accent, I'm not from Canada, I'm actually from Scotland. And I studied at the University of Glasgow, and in my PhD, I made molecules, big molecules that targeted and bound to small phosphate ions. And I really enjoyed doing it because I got a kick out of making something that nobody else had made. And when I moved to Yale, University of Glasgow has been called Hogwarts quite a lot. When I moved to Yale, my supervisor charged me with developing inhibitors of STAT3 for the purposes of killing breast cancer. And for me, this was quite a transformational event because for the first time ever, I was making molecules in my wee round bottom flask in my fume hood for the purposes of killing cancer. And it's the first time that I've ever seen something that I've made kill cancer. And this was transformational, and actually it changed the whole direction of my career towards becoming a medicinal chemist and an anti-cancer researcher. So I've been working on STAT3 for 10 years now, but I think we'd possibly go back a little bit further. I'm from a little island on the west coast of Scotland called Bute. And if you look at the shape of Bute, it looks almost identical to STAT3. So maybe it was fate that I was meant to target this protein. So I'm going to tell you a story today about making drugs, legal ones, not like Breaking Bad, where Heisenberg makes crack cocaine that's 95% pure. I can tell you that my graduate students here at UTM could make crack much purer than Heisenberg. But they would never, ever do that. Ever. So I'm going to tell you today about our efforts to target STAT3. STAT3 has been coined a master regulator of the cancer cell, and it's been well touted as being possibly a transformative drug in the fight against many different human cancers. So the first question is, why do drugs work in the human body? And to answer that question, you have to think of yourselves at the highest echelons of educated society. You have to think of yourself as an organic chemist. And organic chemists see the human body different to clinicians. We don't see it as muscle, fibre, bones and tissues. We see it as a large reaction vessel with big and small molecules that are interacting with each other and undergoing chemical reactions to facilitate our bodily functions. And if you look at it, this small molecule here, cis-retinol, it flexes to allow you to see what's on the board today. Serotonin, a small neurotransmitter, bridges the gaps between your brain cells to allow you to hopefully understand what I'm saying today. The take-home message is that drugs interact with molecules inside your body, inside cells in your body, to elicit the effects that you see in drugs today. So I target cancer cells. What is a cancer cell? What defines a cancer cell? Cancer cells have uncontrolled cell growth. They have uncontrolled cell division. They are champion invaders. They can move throughout the human body and implant themselves in foreign tissues. And they're mortal. They do not respond to all the natural cues to die, and they persist. Why does a cancer cell work? Why does it always want to grow, divide, expand, and invade? It all comes down to the genetic programming of a cancer cell. And the genetic programming of a cancer cell is to grow, and to divide, and to expand and invade. But this is controlled by cell signaling pathways, intricate and beautiful cascades of protein-protein interactions that lead to genes being produced that tell the cell what to do. And in cancer, these genes are telling it to survive and to grow. So what do we target? We went after STAT3, this protein that resides in the cytosol. STAT3 expresses genes that lead to division, growth, preventing cell death, and tumour formation. So how can we target STAT3 if it's already active, if it's performing a normal role in normal cells? Well, in normal cells, STAT3 is only switched on for a very short period of time, and then it's very quickly shut down. In cancer, it's permanently switched on, and because it's permanently switched on, you have this overexpression of genes that lead to the cancer cell. So we want to use medicinal chemistry, and we've been using medicinal chemistry to develop drugs that target STAT3 in the cancer cell. What is medicinal chemistry? Technically, it's the molecular bridge between biology and the clinical science of medicine. From a simplistic perspective, it is the design and synthesis of small, typically organic molecules that bind to a specific protein with exquisite selectivity and potency. And the result of this binding interaction is a biological response. And in cancer, that has to be cell death. We want that interaction to lead to cell death. We want it to be potent so that we use less of the drug, and therefore there is less toxicity if I'm feeding someone a kilogram of drug. And we want it to be selective because if a drug binds promiscuously to every cell in the protein, every protein in the cell, there's going to be side effects, right? Because every protein performs a role, small or large, in the cell. And if you're affecting all those proteins, you're going to have toxicity. So why is it selective? A drug reaches every major organ within 4 minutes. A typical drug dose is 1 milligram. That's 10 to the 18 molecules. So that's a lot of molecules going into the body. Unfortunately, the human body has 10 to the 14 cells. Each cell contains 10 to the 10 molecules. Each drug molecule therefore sees 1 million different targets. So target specificity is hugely important. When your molecule binds to your target, you want it to stay there and elicit that biological response. In our case, STAT3 is a weird one. The active form of this protein is actually a dimer with itself. So there's actually 2 STAT3 proteins here that you can see bound together. And what we want to do was to make a molecule that would bind to it and rip open this protein dimer complex. So how do we do that? How do you design a small, tiny molecule that disrupts a massive, relatively speaking, protein-protein complex? So we took our inspiration from nature, and we mimicked this yellow part of the protein shown here, which helps facilitate this protein-protein interaction. And we used nature's own design to inspire the design of our own small drug-like molecules. So, what is medicinal chemistry in simplistic terms? It's a bit like a jigsaw puzzle. We try to find the molecular pieces that fit in the protein puzzle. So with STAT3, we saw 3 main binding sites in the active site of the protein. And we wanted to make a molecule that could access all of those sites. So we used computational modeling to identify molecules that could bind. And someone found a molecule, and it's called S3I201, and it bound. And you can see that it kind of binds in two of the main pockets, but not in the third pocket here. So what do? So the number that you see here, this IC50 is the concentration of drug required to kill 50% of cancer cells. So 90 micromolar is a very small quantity of drug, but if you want to be really drug-like, you need to have the number closer to 0.5 to 0.1. You need tiny, tiny quantities of drug to kill your 50% of the cancer cells. So we had an idea, and our idea was, well, we're going to build off of this molecule, and we're going to occupy this third pocket. And we need a component that's roughly this size. So we functionalized our molecule with benzenes. Do you remember benzene from your organic chemistry classes? We decided to functionalize it with substituted benzenes to access the upper pocket of this protein. Biologists don't like us. They call us cooks and chefs, and Breaking Bad has propagated that idea when they say they're cooking, but they have an inferiority complex. And this is because chemists can make molecules that even God hasn't made, like Viagra. We have the power to turn old men on, so that shows you how powerful we are. So the first step in our MedChem program is to synthesize a large number of molecules. And we do this using synthetic organic chemistry. So we make a library of molecules. And I'm just going to denote them as these little shapes on the bottom right of your screen. Then what do? We then check to see whether it binds to the protein. The protein outside of the cell, not inside. If it doesn't bind outside the cell, it's not going to bind inside. So we screen these molecules against the protein. And what we're looking for is a number of molecules, or even one molecule, that binds really potently. Then what do? We then test the molecule against the other proteins. Does it bind promiscuously? Our hope is that it doesn't, that it binds selectively to our target protein. Then we target cancer cells. So in this case, these are breast cancer cells, and we treat these cells with our molecule, and what we want to see is that our hypothesis is correct, and that by targeting this protein, the cells die. So if that's the case, we then test against normal cells. In a cancer patient, most cells are normal, healthy, functioning cells, and we don't want our drug to kill them too. And so we want to see a molecule that has a good therapeutic window against cancer cells and normal cells. Then we have to do cell biology. And what we do with cell biology is we look to see whether inside the cell, if our protein is being inhibited. So in this case, you can see your STAT3 protein here, and with increasing concentration of drug, you want to see the protein get knocked down with increasing concentrations of your inhibitor. Then what do we do if that works? You've guessed it, we look at selectivity again. Does it hit everything inside the cell? We look at other proteins to see whether there's an effect. And ideally, we're wanting molecules that don't have an effect against other proteins. And the final stage is preclinical for us in academia. We look at human tumours engrafted in mice, and we're looking to see whether it kills the tumour and whether there's toxicity. So what have we done at UTM, at University of Toronto Mississauga? We started off with this compound that was 90 micromolar. Remember, we're trying to make it a lower number. The first modification that we made, as you can see, really badly affected that number. It went higher than 300. So we lost all activity, but we kept with our program. We realized that we had to functionalize this pocket in order to get activity. So we put on a benzyl 300, naphthyl 280, paracyano 260, butyl 194, biphenyl 115, cyclohexylbenzyl 4. So this was 20 times more potent than the original compound. Being chemists, we wanted to get that number lower, so we optimized again, And what we identified was this molecule, BP1102, which was 0.8 micromolar. So this was a really good starting point for us. It's 110 times more potent than the starting material. And what we found in non-STAT3 cells was that it didn't kill those cells, which was great. And what we found in breast cancer cells was that it dramatically affected the number of breast cancer cells, killed them. We then looked inside the cell. Did it affect STAT3 inside the cell? And what we found with increasing doses of drug was that we almost completely inhibited STAT3 protein inside the cell. Then we put it inside mice, and we injected it inside mice, and we looked to see what happened. So you inject it into mice, it goes through the liver, and guess what happened? This. Our molecule quickly turns into this molecule. Now think about the jigsaw puzzle. Will this molecule fit in our jigsaw puzzle? No, not at all. So what we actually found was that the drug disappears within 10 minutes inside a mouse. So there's no effective dose of our drug there. So what's the solution? Chemistry. We had to go back and we had to fix the parts of the molecule that were not stable. And what we did was a chemistry program where we changed the structure. And the structure changed, but its affinity to the protein did not. And we ended up with this compound, AM1124. And AM1124, when we injected it into mice, unlike BP1102, survived for almost 4 hours, which was fantastic. So it now meant we had enough drug in our animals to see an effect against tumours. Then we did a wee bit more chemistry, but it's patent protected and it's a bit of a secret, so we can't actually tell you the structure. And we're going to call it Compound X. So what we found with Compound X in preclinical trials against HER2-positive breast cancer, we were looking to see whether the breast cancer metastasized to the liver. And what we found was that in animals that were not treated with the drug, 80% of the mice had tumours, secondary tumours forming in the lung. With our compound, less than 10% had tumours. And this is a really cool image. This is one of the coolest images of my career. I'm going to tell you on the slide it's a cool image because probably our definitions of cool are pretty different. But this is the tumour in mice, and it's glowing green because it's got a marker in there that shows STAT3. And what we found in a very short period of time is that we were shutting down STAT3 in a human tumour in a mouse, which we thought was really cool. We also did brain cancer, and you can see this is a slice of the brain with the actual tumour stained in white. When we treat with this molecule, we saw really pronounced, really beautiful effects against glioblastoma brain tumours. And I'm going to show you one last trial with this molecule. This is in medulloblastoma. These are chemotherapy and radiation-resistant children's brain cancer tumours. And what we found, and this is mice models, this is their brain in purple, you can see the brain tumours. When we treat with our molecule, you see really quite stunning regression of these tumours, so we're really excited by this study. And I want to finish by just summarising. We started off with a molecule that had lots of different targets and wasn't a particularly good drug, and we optimised in a very rational way towards Compound X, which we believe is on target. Maybe. There's a million different targets, so it probably is hitting something else, but we're really excited by the initial preliminary data. And I'd just like to thank my research group, who do all the work in my lab, and all my sponsors who help do this research. Cancer research is extremely expensive, and I've just got time to show you one last thing, the power of organic chemistry. In 2007, we started off with 600 square foot on the 5th floor of the William Davis building. Then we got some money, and we got a bit of a lab in the 3rd floor. And then we continued to expand a little bit more, needed some office space. We decided in 2014 to actually do cell biology, and we built a cell biology lab. So if you've watched Game of Thrones, chemistry is coming. Thank you very much.